Hello, I'm Michelle Werner. I'm the CEO of Alltrna. What makes engineered tRNA therapeutics fundamentally different from other RNA-based therapeutics or genetic medicine approaches? Well, most genetic approaches take a single drug, single disease paradigm because they treat a certain gene or a certain protein. Engineered tRNAs are fundamentally different from all other genetic modalities because it's the only one that a single engineered tRNA may be able to address hundreds, if not thousands, of different diseases. How can tRNAs achieve a shared mutation approach? Well, what's really special about a tRNA is that instead of focusing on a gene or a specific protein, which is what all other genetic medicines do, a tRNA looks at the mutations that are common across many different diseases. And by addressing the mutations that are common across genes and diseases, what we may be able to do with our engineered tRNAs is to allow for therapeutics that can treat hundreds, if not thousands, of different diseases. By doing so, patients who have one of these genetic conditions, maybe up to 10,000 different diseases, instead of waiting for individual treatments for each and every one of them, patients across thousands of life-threatening conditions may be able to be addressed with a single engineered tRNA. Why are premature termination codons the right place to start? Well, a premature termination codon occurs when you have a code for an amino acid codes for a stop instead. Now, there are no tRNAs that know how to read these premature termination codons, and therefore protein translation terminates too early, and the result is a truncated or dysfunctional or oftentimes absent protein. By addressing premature termination codons with our engineered tRNAs, we can read through those premature termination codons and restore the protein translation process and restore the production of full-length protein. Now, this is really important because there's a fundamentally specific genetic cause of these diseases, and by addressing premature termination codons, that clear biology can be demonstrated both preclinically, as we've seen already, and hopefully translate into the clinic. How have engineered tRNAs been de-risked as a therapeutic approach? Well, our engineered tRNAs have been in development from a preclinical perspective for over the last 5 or 7 years. We've made a tremendous amount of progress really demonstrating in an in vivo capacity their ability to restore the production of full-length proteins across a number of different disease models. We also have a very robust safety package that demonstrates the safety of these engineered tRNAs, which is really important as a new modality. And our engineered tRNAs, and specifically the first engineered tRNAs that will be advancing towards the clinic, are formulated in lipid nanoparticles. They allow us to have the ability to have tunability, to redose, to allow to adjust those doses, and to have reversible effects if necessary. So it really is a de-risked option, even for a brand new modality. What are the major milestones for Alltrna in 2026? What we're really excited about this year is that we're entering into the clinic. This will be the very first time that a tRNA, an engineered tRNA, has been given to humans. And it really establishes the modality in a very pioneering way to really demonstrate that what we've seen in an in vitro and an in vivo perspective actually translates to humans as well.